So I'm going to tell you this story that we published last year and that has some commonalities with things that have been discussed in the meeting. So the main issue that we are interested in this work is kind of basic questions in sensory coding. And they start from the observation that you're all familiar with, that the responses of sensory neurons are variable. Okay, so you present some stimuli in this example. So this is an example from visual cortex. You present oriented gratings, and you see that the stimulus, the neurons care about the stimulus. So if you plot their responses to different features, they do different things. But the stimulus only captures a moderate amount of variance in the response. And so if you plot the responses, in this case one neuron versus another neuron, we can't really see very well. But anyway, so maybe I have to use one hand with this and the other hand with this. So if you look at one neuron versus another, and this is each of these plots is one condition, you see that there's still a lot of variability. And this is quite typical. It's not universal, but it's very typical that typically the thing that the experimenter controls only has access to some finite amount of the variance. Okay, and so this, if you're thinking about someone reading out the activity of this neuron, this poses a problem because maybe the stimulus was this, and this neuron on average says this, but sometimes it says this, and so then you might wonder whether then the stimulus is this one or this one. Okay, and so there's been a lot of work for decades from computational neuroscientists, mainly trying to understand how the shape of the noise affects the way the stimuli are represented and the ability to decode the stimulus, etc., etc. So I highlight here just two papers. One, this is from Stefano Panzeri 20 years ago, and they were one of the first ones, I think, that very clearly made the point that the extent to which the variability matters depends on its relationship to the trial average responses, so that the same kind of variability can be harmful or not from the point of view of decoding or information, depending on how it relates to the signal. So for instance, in these two cases, variability is similar, but the signal is structured differently. And so they have very different consequences in terms of how the population of cells represents the stimulus. Perhaps a simpler way of showing it is here in this review, where you see again the activity of one neuron and another. The dots here will be the trial averaged responses of these two cells, and the ellipses indicate the trial-to-trial variability, the kind of coverage of the trial-to-trial variability. And in these two cases, the shape of the trial-to-trial variability is the same, but intuitively you see that here it doesn't hurt and here it hurts. And that's because in this case, the kind of orientation of the noise is similar to the orientation of the signal, whereas in this case, the signal is varying in this direction and the noise is varying in this direction. Okay, so this is kind of the essence, I think, of all this work on noise and sensory coding is that situations where the variability generated by the noise and the variability generated by the stimuli are exploring similar regions, then that's going to be problematic, whereas if they explore different regions, things are okay. Okay, and so the other angle of this presentation, which is something we're very interested in the lab, is that all these properties are dynamic. They change and they depend a lot on behavioral context. And so one aspect of behavior that we study is what's called associations, the changes in dynamics associated to brain state. And so I highlight here the work of van der Wolf. So this topic has kind of become fashionable again for the last maybe 10, 15 years. But actually there's a lot of work in the 70s and 80s by him and people like him who were very interested in just looking at spontaneous behavior in rodents. And they were recording LFP or EEG kind of coarse measures of neural activity during different behaviors. And they found that there's a big dichotomy, a change between what they call kind of voluntary behaviors. So they mention walking, swimming, running, digging, orienting, rearing, all these things. And what they called automatic or type 2 behaviors, licking, grooming, chewing. And so basically they're making the point that there is two things that seem to affect the dynamics of the brain. So you see here, this is the signal, and this is when different behaviors, type 2 or type 1, in the cortex. Here you basically don't see any low frequency oscillations. Here you see a lot. And in rodents, it happens that this correlates with movement, because as soon as they start running, you see something like this. But they make the point it's not just movement, okay? Because there's a lot of these automatic behaviors which involve movement, and still the cortex is synchronized, and you see theta in the hippocampus. And so this should be interesting to all of you, because as I'm going to show you, it's kind of the main, it completely reconfigures the dynamics of the population and the way the signals are represented. So it's a very big signal. It's the order 1 effect on activity is associated with this change in dynamics. So it's observed across many different conditions. I just talked about wakefulness. So there's this kind of attentive versus unattentive quiescence, locomotion, whisking, immobility, okay? This axis of engagement and axis of movement. But it's also observed during sleep. Where you have REM, which is activated, and slow wave, which is inactivated. And also under anesthesia, and the experiments I'm going to tell you today are under anesthesia. So there's this particular anesthetic called urethane that targets the cholinergic system. And it's very nice because under urethane you get both extremes, okay? So you get both extreme activation and extreme inactivation. This is another example of already, not so recent paper, but very influential, which was one of the papers that kind of reignited interest in state, where you see, in this case, two neurons and how their correlations change very much. During quiescence, they're strongly correlated, whereas during whisking, they're uncorrelated. And we also contributed to this, looking at things at the population level, where you see, again, this during inactivation or synchronization, you see these global fluctuations in firing rate, so they're very salient. And during activation, it looks kind of like a balanced network, so you don't see much, okay? So today I'm going to be talking about how, when things look like this, how the network represents stimuli compared to when they look like this, okay? And it's important, I'm going to be mainly referring to it as activation, inactivation, even though it's not inactive. I mean, there's spikes, okay? In fact, there's not very different numbers of spikes in both cases, but just the convention is to call this inactivated. And also, I'm talking about kind of long time scales, okay? During inactivation, you have alternations between periods of activity and periods of silence. But in general, this is a single state, okay? This would be an up state and a down state within a period where brain state is inactivated. Yeah?
So are these two distinct states, or is there a continuum?
No, it's a continuum. So it's a continuum, yeah. And so what stimuli are we going to use? So we're going to study the representation of lateralization of intensity and overall intensity. And we do that because we're interested, so we train animals to do tasks where we make these headphones for rats, and we play a sound here that is slightly louder than a sound here, and they have to tell us which side is louder, okay? And so we're interested, in general in the lab, quite interested in how the brain deals with intensity. And in particular, this preparation is very nice because you can very the overall intensity in the two sides or the relative intensity. Okay? And this is kind of very ethologically relevant behavior because the difference in intensity is the cue that rodents use to localize sound in the horizontal plane. We also use it, but we also use timing differences, but they use mainly intensity differences. Okay. Yeah.
Do you also calculate the threshold for every mouse separately? Because there seems to be quite some interaction between frequency and intensity in mice.
Yeah. So you're right that this signal, it's called interoral level difference, ILD. So I'm going to be talking about ILD all the time, which is, so if you have sound in one ear and sound in another ear, ILD is moving in this direction, and ABL, absolute binaural level, is in this direction. Okay? So ILD is strongly frequency dependent. Okay? It's a signal that is higher in the higher frequency. So you can see it here. You play sounds, and then for the high frequencies, it's very different between the two ears, whereas for the low frequencies, it isn't. The sounds that we're going to use are broadband noise. Okay? So they're 5 to 20 kilohertz. So there's no frequency component in the sounds that we use, but for the animal, it's very important. So yeah, we study basically how they do this, and there's some nice kind of phenomenology that basically the accuracy with which they distinguish ILD doesn't depend on overall intensity. So you see here the psychometric curves for quiet sounds or loud sounds are the same. Okay. And so we started doing these behavioral experiments, and we were curious about how these signals would be represented. And so that was the motivation for the recordings. So we record with this kind of electrodes. We put them here. Okay. In these experiments, we come in like this, tangentially to the cortex. So most of the data comes from the deep layers. Okay, probably layer 5. And because there's all these contacts that are close together, they're quite good for spike sorting, and so we record typically 100 neurons or so simultaneously on each recording. This is the stimulus set, again, which is very similar to the one we use for the animals, except that we include a couple of more extreme cases because the neurons are typically broadly tuned. And then we present sounds for 150 milliseconds, wait 850 milliseconds, and then the next second we play another sound. Okay? And so it's very systematic data set. As I said, this is the stimulus, duration 150 milliseconds, there's 36 conditions, so 3 loudnesses and 12 lateralities, and there's 100 repetitions of each of these 36 conditions. Okay? Yeah, it's a nice, and the data set is on the order of 20 sessions, average around 100 neurons per session. And so again, when you stick your electrodes, you see these kinds of transitions, okay, between active state and inactive state, and we're going to quantify state by, you can do it several ways. The way we're going to do it typically is just looking at the CV, the coefficient of variation of the MUA, okay? And so in this case it's small, in this case it's big. You can also look at density of downstates, but in this study we focused on the CV, okay? And this was just to show you that actually in this paper it was very nicely characterized that the phenotype of transitions that you get between activation and inactivation under urethane is identical to sleep. Okay? So you can think of this as a fairly accurate model of transition of state during sleep between REM and slow wave sleep. Okay, and so the goal in the task is to evaluate the structure of the neural code at the population level, and we're going to do it kind of systematically. First, we're going to look at the global activity, the MUA, and so this is essentially the projection of the population activity to the uniform vector. So this has come up a couple of times, and it's going to turn out to be quite important. Then single cells, then we're going to look at kind of population level features, and then we're going to study a bit decoding. And the main goal is to try to understand how it changes across brain states. So first, the multi-unit. First in the inactive state. So this is one particular recording. then if you look at for that stimuli, you choose one particular one. This is one neuron, okay? The repeated presentations. This will be another neuron. These are all the neurons in this recording. So the first thing we're going to look at is just their average, okay, the MUA. Oops. Yeah, so that's the thing that we're interested in at the moment. And that was for that stimulus. For the 6 stimuli, it looks like this. And so we can just collapse them. You can see them there. So these are all of them, okay, for the 36 conditions. In general, we're always going to use the same color code, okay? So blue to kind of orangey is lateralization. This is contralateral, and this is ipsilateral, okay? So the recordings are made in one hemisphere, and blue means the sound was louder in the opposite hemisphere, okay? And then the size of the lines or all of the dots encodes overall intensity, okay? And so this is one particular session. Then we're going to be bold and just count spikes, okay, for the duration of the stimulus presentation, which is 150 milliseconds here. And we're going to get then tuning curves. So this is what's a tuning curve for us, okay? It's 36 numbers, which is the mean response, in this case of the MUA, to the 36 stimuli. So this is lateralization, and each of the 3 is one intensity, okay? And so here you see what turns out to be very typical in the inactive state, which is the textbook, what you would expect. The neurons, and in this case the MUA, prefers contra, okay? So blue is always bigger than orange, and it also prefers loud to quiet, okay? So maybe that seems kind of obvious. We're going to see it's not universal, but that's what you see in the inactive state, okay? So the population rate prefers loud contralateral sounds. Now, if we look in the active state, which has this other kind of phenomenology, then we see it's quite different, okay? So these are the MUA for this particular session, and you see that it's actually fairly flat. I didn't say, but the small dots is if you just count spikes in this period here, the onset response, okay? So you see it's fairly flat, and it's especially flat in the middle. The middle is what matters, okay? It's the situation where the sound is either here or here, and so behaviorally that's the relevant region. Rodents are not good at telling this from this, but they're quite good at telling this from this, okay? And so you see that even though you're sweeping contra to ipsi or quiet sounds to loud sounds over kind of 40 dB, which is quite a big range, the auditory cortex doesn't seem to emit more spikes. So we just quantify this by, say, taking what you expect to be the highest stimulus, which is this one, contra loud, and we divide by ipsi quiet. Okay, so that should be the highest contrast. And so you're going to see a lot of plots like this in the presentation. Each dot is a session, a recording. Okay. Then there's 2 dots that are always in a circle, and those are our highlight sessions. So we just pick 2 experiments, okay, and you're going to see the ones with a circle. And then we have 3 dots in one color, which are the sessions that we kind of arbitrarily defined as active. Then these other dots are the ones that we define as inactive. But all the summary statistics are always regressions on CV, okay? So we don't, and that's getting to your question, as you can see, it spans the whole axis, okay? And we don't make qualitative distinctions, we just see whether things change with the level of synchronization or not. And so in this particular one, what you can see is that contra loud to ipsi silent is effectively 1 in the more active experiments, whereas it's quite big in the inactive experiments. Okay, so that's that for MUA. What about single cells? The kind of puzzling result that you don't see more spikes for louder contralateral sounds might be because you don't see tuning, basically might be because the cells are not tuned, or maybe they're tuned, but they're tuned in a way that is kind of orthogonal. Okay, so that to distinguish that, we look at the cells individually. So again, these are the PSTHs for each condition for one particular cell. Okay, and so we again count spikes during the stimulus, and this is the tuning of this particular neuron. So this is more like a textbook neuron, again, fires more for contra, loud sounds. This is another neuron, this one's different, it prefers quiet sounds. This one prefers contra sounds. These are a bunch of them, and so you see that there's a lot of diversity, and that's very standard. I would say that's the default in cortex. But we see it mainly during the active state. Okay, so we see neurons more like the textbook, like this one, but this one is more like anti-textbook. Okay, so you see it's exactly opposite preference. So the representation is very diverse at the single neuron level. So how do we quantify it? The way we quantify it is we regress the C-scored firing rate on 3 regressors. Okay, one is ILD, so lateralization. Another one is overall intensity, and we subtract 40 because we want each of the regressors to be centered. Okay, and then we have an interaction term, because conceivably you saw maybe in some of the plots loudness might change the way lateralization affects the neurons. Okay, so we quantify that with this third regressor. And so what do we see? So this is for this particular neuron, for instance. First I'm going to focus on their dependence on intensity and lateralization. So then you will get a point here. Okay, so neurons, these 2 coefficients live somewhere in this plane, and this is this particular neuron. These are all the neurons in this recording where I took off the neurons that are not tuned. So those are the ones who would live here. But you see that it's nicely symmetric. Okay, so it seems like the representation in the active state is symmetrical with respect to these 2 features. Okay, even though it's a single hemisphere, everything seems to be somewhat equally represented. These are our 3 active experiments, and this is just a histogram of the angles so that you get a sense of how things fall. It's quite different from the inactive state. So in the inactive state, these are just a bunch of neurons from a recording, and you see that they tend to be all, as we saw in the MUA, contra loud preferring. Okay, so then when we do the same analysis, we see that basically neurons are in this quadrant. Okay, and you can also see it very clearly there. So the inactive state is more kind of like the textbook notion. There are you can find papers who say rat auditory cortex doesn't have a single ipsilaterally tuned neuron. So we see that's fairly accurate in the inactive state, but in the active state is very symmetrical and diverse. Okay, and again, you can quantify it by looking, say, at the fraction of neurons with contralateral or with loud preference, and you see that both of those increase with the level of synchronization or inactivation. So what about gain? So gain is kind of interesting. So what we plot here is the kind of interaction coefficient versus the lateralization coefficient. So what do you expect from a standard gain modulation picture? So let me maybe bring back the plot where this was more salient at the level of the MUA. Yeah. So you see it here, right? The slope of this is smaller than the slope of this. Okay, so the lateralization coefficient, in a sense, measures the slope of this. The question is, does it increase with volume? Okay. So if neurons are gain modulated in this way, what you expect is a positive correlation between the lateralization coefficient and the overall the interaction coefficient, right? So a contralateral neuron would be more contralateral the louder the sound. And so that's, in fact, what we see in the inactive state. Here, you see? So this is the lateralization coefficient, the interaction coefficient, and more dots are here. Okay? But again, it's not really what we see in the active state. So in the active state, we see mostly uniform distribution. We quantify it by looking at fraction of cells in these two quadrants. Okay? And we see that again it starts at 0.5, meaning not much going on, and then it becomes more clustered as it becomes more synchronized. So this very kind of salient feature of cortical processing, you see it also in the visual domain. For instance, when you do tuning curves to orientation, you see that overall contrast increases the gain of the tuning curve. We see that that's typical behavior in the synchronized state, but in the desynchronized state, there is gain modulation, but it's happening kind of idiosyncratically. Okay? So it's not that the coefficient is 0, but it's not operating at the population level in the direction that you would expect. All right. So now the population level, again, you're all very familiar with this kind of way of looking at things. So we're just going to look at the firing rate of the population as a point in a high-dimensional space. Okay? And so this would be, for instance, different presentations of a single sound, and this would be the mean. Okay? So this would be the population PSTH. The population spike count for us is just a point. And then you might have other stimuli here. And so the signal subspace is defined by kind of the region in space which spans more variation at the level of the mean. And then you also have a noise subspace, which is, for a given condition, what's spanned by trial variability. Okay. So the first thing we do is we just look at the signal and noise correlation matrices. So these matrices have dimensions equal to the number of simultaneously recorded cells. And this particular one, the signal correlation, each dot, when it's orangey, means that the tuning curves, which are these 36 dimensional elements, are very similar. Okay? And so what you see in the inactive state is consistent with what we saw when looking at the cells. There's like a big cluster here, okay? And these are the cells that prefer contrast. So we've sorted them according to their lateralization coefficient from the linear regression, okay? And so you see that many cells look like this, and that's why you see this cluster here. In the active state, on the other hand, we see something that is like this, okay? So we see cells that are contra-tuned, cells that are ipsi-tuned, and of course between themselves they're negatively correlated at the level of the signal. Okay? And we can do the same thing at the level of the noise. And what we see is that in the inactive state, noise also is correlated across neurons which have this preference. Okay? Whereas in the desynchronized state, there's not much going on. Okay? So you don't see a lot of structure in the correlation matrix. So how do we quantify it? Yeah.
It's like in the noise case, in the inactive one.
Sorry.
You're pointing at the top left corner, but there's actually then correlation of those with some of with what I guess are the ipsi tunes cells as well, right? So it really looks like there are yellow stripes running both horizontally and vertically.
Yeah.
In that corner. So I'm just wondering whether some of that is being driven by firing rates and noise amplitude effects, right? So if I'm actually all correlated, the whole system's just going up and down, as you might expect in an active state.
Yeah.
But I have different firing rates. I wouldn't see that correlation in a low firing rate cell. I would see it more in a high firing rate.
Yeah. Also, there's very few neurons that are tuned to ipsi.
Sure, okay.
So I don't, so this might be very quickly firing cells, actually. It's cells that have a small coefficient, but it's probably not -1. It's probably not negative. It's probably to 0. Because we didn't see a lot of cells like that. Yep.
I'm sorry, I'm a bit obsessed, but this is following up on Manish. So this sort of suggests that the noise correlation is low rank, because you have this
Yeah.
Is that,
So we're going to look at the dimensionality. Yeah, we're going to look at the dimensionality of the and the orientation of these subspaces. Yeah. So yeah. So then, if you look at the spectrum of these matrices, then in general the signal is lower dimensional, as you would expect. Okay. We quantify it in a not very sophisticated way. We just shuffle neurons. So we do kind of Monte Carlo shuffling of the actual spike trains, and then we take a 99% confidence interval, and we look at how many eigenvalues go above it. Okay. Which is a conservative measure, actually. And so you see that for the signal, average correlation, which is the mean of the elements of the matrix above the diagonal, increases, okay, with synchronization. The fraction of variance in the first principal component also increases, and dimensionality with this criterion decreases. Okay. For the noise, so you see here, actually, we're not even getting the main difference is this versus this. But since we're here just counting how many are above, then we're getting big numbers, okay? And again, you see effectively the same tendency as with the signal. So more correlations, more lower dimensionality in the sense of higher variance in the first PC, and lower dimensionality just counting how many go beyond the shuffled null hypothesis. Okay. But again, this doesn't look at the relative orientation of the manifolds. Okay. So for that we go again to pictures like this, and we're gonna also introduce or pay attention to this global direction. So the global direction is simply the mean activity across all the PSTHs. Okay. So it's effectively the dimension across things are fluctuating during the up and down fluctuations. Okay. So there's these three kind of subspaces of interest. This is a single dimensional one. This is where the signal lives and where the noise lives. Okay. And so I think the first thing that I have is a video showing what we do is basically we do PCA first averaging across lateralization, okay, and then we get the first axis for overall intensity. Then we average across overall intensity, and we get the first axis for lateralization, and we define the subspace spanned by those two vectors as the signal subspace, okay, which themselves might or might not be orthogonal, okay. And then in the animation I'm going to show you, we have the subspace generated by those two vectors and the global, okay. So it looks like this for the active state. And so it's very nice. It's another one of those examples where basically the representation is isomorphic to the stimulus, okay. So if you remember, the stimulus set was also these three rows, and so that's how things look in the active state, where again this is the global direction. So you see that the signal subspace is kind of orthogonal to the uniform, and it resembles the stimulus set. Whereas during inactivation, then the three subspaces kind of align to each other. So you have both the signal, the noise, and the uniform become more compressed. Okay. So this is for two particular experiments. Again, the signal subspace divided into the ABL axis and the ILD axis, and then we're projecting both the mean and the trial-to-trial variability, and these ellipses are, I think, 50% coverage in the plane. Okay. And remember that this is not actually the direction of the noise. The direction of the noise is probably living somewhere outside of the screen. Okay. And you see the kind of really striking difference between the active state and the inactive state, where here you're just effectively getting a copy of the stimulus set. Yeah?
Does this reflect also in the behavioral performance?
So I'm gonna, yes, but for the wrong reason. I'm gonna describe it, yeah. Yeah, so maybe you're hinting that this organization would allow you to do level invariant decoding. Yes, it does, but that's not how the rats do it. But we look at it. So here basically we again quantify this by looking at the angles between all these subspaces, okay? So between the two signal directions, the noise and the signal plane, the uniform and the noise, and the uniform and the signal. And you see that they basically all kind of go down with synchronization, okay? So everything is kind of organized around the mean activity, but only in the inactive state. Okay, so finally looking at decoding, again, because we actually have these animals doing this, then even though this is activity that is not recorded when the animals are behaving, then you can ask how well could you actually infer the lateralization of the stimulus from looking at the population. Okay? And so the way we do it is we use logistic regression to predict the lateralization, and the lateralization is the sign of ILD. Okay? So it's whether you're on this side or on this side. Okay? Using the population activity on every trial. Okay? And just for the aficionados, we use kind of elastic net regularization and use nested cross-validation to test the accuracy of the different models. And so this is what you see. This is for one particular experiment. Okay? These are what would be the population neurometric curve. Okay? For the three different volumes. Okay? So here is lateralization, and each of the three intensities is one of these curves. And as you were hinting before, we see that in the active state they're very similar. And in fact, this is using the same decoder for all the volumes. Okay? So not only the information is there, but actually the same decoder can effectively recover the performs equally well at distinguishing regardless of intensity, whereas in this case is very clearly not the case, and overall intensity makes a difference. Okay? So you do better for for more intense sounds, and worse for quieter sounds. And again, this is reminiscent of what we saw with the behavior. By the way, this is because this is in dB, this is Weber's law, right? This is telling you that accuracy just depends on the ratio of the intensities. So this is something we're really interested in. And these are, so it turns out that we actually understand quite well how it happens behaviorally. And it doesn't happen like this because behaviorally there's a really interesting reaction time correlate to this. So even though the accuracy doesn't depend on intensity, reaction time strongly depends on intensity. Whereas this solution just simply suggests that you get rid of intensity altogether. And so in this case, we know that intensity is still driving the behavior because the animals are faster here than here. Whereas here it doesn't, and in fact when we look at kind of decoding as a function of time, we don't see the same pattern with reaction time with this data that we see with the behavior. So there's like two independent ways of doing this. One is basically just organize intensity and laterality orthogonally, so you can decode one independently of the other. And then there's another way that it's another talk that has to do with intensity actually not having an effect on accuracy, but determining reaction time.
Yeah, but no, I was just actually you just summarized it. So if I understood correctly, the reaction time itself could be just an orthogonal subspace, right? And they could be making their estimation through this kind of invariance.
So it could happen in principle. It's just that a model where you assume that the neurons actually are still kind of overall volume dependent, so that the representation of laterality changes with intensity, predicts trial by trial reaction time in a way that is really accurate. And so it's hard to believe. I mean, it's a model, but it's a model that for kind of psychophysical standards is very accurate. And so it suggests that the two things are really intertwined. You know what I mean? Yeah. In fact, it's fairly deep question, because so this finding is old and it's generic. I mean, it's Weber's law, so it's very general. But even in this particular context of ILD lateralization, has been seen in many species, and the way people have been looking for it at the neural level is trying to find ILD kind of level invariant representations. Okay, so that would be something like this. But we're saying that you can get invariances in the behavior that are not in the neurons. So if you look at the model that actually produces this, there's no neuron in there that has this invariance. Okay, it's purely a computational property, and that I think is quite important. That
comes out only at the population level.
No, not only at the population. There's no single there's no static decoder of the neurons that has this invariance. It's a computational property. It has to do with bound cross, the activity crossing a bound, but there's no neuron in the system that has this property, that has the same tuning regardless of overall intensity. Not even at the population. There's no decoder that has the invariance. It's a computational property. And that's not, so here we see that actually it's just the invariance is there at the neural level. Yeah, and so basically this is just how we quantify the effect of accuracy. So we have two things we care about. One is overall intensity, and the other one is the degree of activation through the CV. And so we fit this model that basically allows overall intensity to change the intercept, and also overall intensity could change the slope. Okay? And so for instance, for 1, these are the dots for the lowest intensity, and you see that things go down, accuracy goes down as a function of synchronization, as you expect. But when you look at all of them, basically the model chooses a solution where they all cross here, again, showing that accuracy is level independent. Okay? So the slope changes, meaning the way accuracy deteriorates with synchronization depends on loudness. But when it's desynchronized, then it's level independent. And then we do you can do all sorts of tricks. So first you can actually fit separate decoders for each overall intensity. And again, you see that when you're synchronized, decoders fit separately do better. But when you're desynchronized, it's the same. Okay? So it doesn't matter. You can do as well just looking at one decoder for all intensities. Then we can explicitly look at the role of the noise correlations in this by shuffling the data. And we see again that here there's no effect, but there's an effect here. Okay? And then we can, if you think about it, this is a point that was made in that paper, that review from 2006, that even though correlations might be bad for information or might kind of limit the information content, it could still be the case that the best decoder is independent of the correlation level. And so to assess that, what you do is you train the decoder on shuffled data, but you assess it on training data. Okay? And here we see that actually when we do that, we see a change again in the inactive state, okay, but not in the active state. All right, so I think that's it. So in summary, in general, encoding is more efficient in the active state. The efficiency depends on an organization where the different relevant features, in particular the intensity difference under sum, are kind of locally symmetrically represented. It's not just the noise. So that's very important. Kind of default assumption is always, okay, so you have this signal modulating everybody, so this is going to introduce noise that is positively correlated along the uniform direction. And that's true, But we see that tuning curves are really strongly reshaped. Okay, so it's actually the way the system is representing the stimuli is fundamentally different in the two states. In the activated state, all the relevant subspaces, so the global, the signal, and the noise, are approximately orthogonal. And so you see this is a bit different from the data that Manish was analyzing the other day, where he was seeing that even in a behaving context, there was significant amount of variance by the population in the global direction. And then in the inactivated state, all the dynamics are kind of structured around the global axis, whereas this subspace is not really prominently explored during the active state. So in a way, you can think of this as saying that in the active state, the cortex transforms an intensity code into an identity code. Okay? I think this is fairly generic of cortical representation, where you're trying to basically get rid of the low-level feature aspects of the stimulus, and just neatly partition everything into a representation that is easier for learning, in the sense that there's different neurons caring about things that might be of interest to the animal. All right, and if you're interested, as I said, this was just published last year, so you can check the details there. Let me just thank so Dimitri, who was a postdoc with Christian, but we worked together for this project, and he did the analysis. Then Jose did the experiments, and he also did all the behavioral work that I didn't talk about in collaboration with Mafalda. Thank you. Yeah.
Could I go back to this question, because I'm not sure I understand. We agree that it cannot be a single unit property. It has, but you said it's not a population property; it's a computational property. Yeah. And I'm not sure I understand what you mean.
Of course, you don't understand, because I didn't explain it. But it's another. So we can talk about it later. It's just okay. The way to explain the behavior of the animals is very beautiful, and it involves accumulation of evidence, blah blah blah. But it has the property, if you look at how the model is solving the task, that there's no invariance in the neural representation. Okay? There's no invariance. There's no decoder, not single neural level nor population level, that is level invariant. Okay. Basically, what happens is that the probability of hitting the bound is a ratio, okay, of the mean and the variance. And when you increase the volume, both the mean and the variance increase by the same amount. And so the probability is invariant, but all the neurons are firing more. There's more spikes. That's why reaction time is shorter, because there's more spikes. You hit the bound earlier. Okay.
So it's like Weber's law, essentially.
Yeah. So what we're saying, we're making this claim that in general Weber's law is actually the reflection at the level of accuracy of another regularity that also involves reaction time. If you just look at accuracy, there's many ways to explain it. This would be, these kind of decoders, or people have come up with many explanations. But when you include the reaction time phenotype, actually, then there's not so many ways to do it. Yeah.
How do you get the data with the behavioral model?
So we don't actually think it's cortical. So this is recordings in the cortex. Okay. We're now basically first doing inactivations and lesions in different stations, trying to understand where exactly is happening. But it seems, so there's prior works showing cortex is not critical, and we've done it ourselves, and it seems like it's not critical indeed. That doesn't mean that it's not critical for our task. That doesn't mean that there may be many tasks where knowing about intensity and about lateralization is important and the cortex is involved. But for this particular task, we don't think these neurons are the neurons that are responsible for the behavior.
This is why you don't see the invariance there.
Yeah. Of course, we didn't know that when we started to do this recording, but...
Did you mention integration of evidence, but the stimulus is 150 milliseconds.
Yeah, because in this case, again, here, this is not a reaction time paradigm. The behavior is very important, that is reaction time. So the stimulus needs to be going on until you make a choice. And in fact, If you now take the stimulus and make it 100 milliseconds, you break Weber's law. So that's really strong evidence that it's a phenotype that has to do with accumulation of evidence. If we had played these sounds, or if we play these sounds to the rats, there's no Weber's law, okay? Because it really depends on you having enough information until you reach the bound.
So how rapid are these transitions between active and inactive states? And do you think this behavior is a consequence of the dynamics, or some circuit reorganization is taking?
No, I don't think so. For the behavioral work, the animals are very focused. We haven't done recordings in those animals, but my intuition would be that things are going to be fairly activated. The transitions, so that depends on exactly what model do you use to study, to explore the continuum. In our model, it's the same dynamics as during sleep, meaning the periods are on the order of tens of minutes, the durations and the frequency of the transitions is very similar to what you observe in sleep, okay? In behavior, is very different. So we're now actually doing, not for these animals, but we do experiments where we train, say, mice to discriminate frequency, and we record, and you can look at the level of synchronization, say, before, the 1 second before you play the stimulus. And you see trial-to-trial variability. It's not so extreme as this, okay? And when you look at the autocorrelation function, it's very, there's a peak at 0, and then there's like a small component, but there's a lot of variability that is happening on fast timescales. And that's consistent with work that some people have done, like trial-to-trial fluctuations in attention or focus are associated with behavior and with this kind of global dynamics when you record population activity.
Thank you very much.
Sure.